So I am from Office of Research Integrity, as Ben told you. I work on oversight review of research misconduct investigations that primarily are taking place in the institutions on public health service funded research. And we mainly work on, most of our cases are NIH funded research. We also work with our Office of General Counsel when there is need for their support in cases of misconduct. And when there are human subjects involved, we work with Office of Human Research Protection, and also work with Office of Inspector General when there are additional concerns other than misconduct. But also when there is joint funding with VA, NSF, and other sister officers, we do collaborate with them when there is a new joint funding. But overall, like today's meeting, our office, ORI as a whole, we work with other agencies and institutions like international universities, medical centers, biotech companies, and so on, by working like a conference today is an example of that. So to improve integrity in research. So with that in mind, and the focus of the conference, I'm going to see if can technology help and improve efficiency and reliability. These are two key components, important parts that we need to focus when we talk about technology in research misconduct investigations. So I'll be presenting the steps that we go through in misconduct proceedings with examples and how we use existing technology or existing tools, and convince you all the real need for technology to help us improve, again, efficiency, reliability. So what are the steps in research misconduct proceedings? You all know that it starts with an allegation, and anyone can make an allegation, and whoever is receiving the allegation will do an initial assessment. Usually it's every institution has who receives PHS funding has a research integrity officer. They do the initial assessment, but if ORI receives the allegation, we do the initial assessment. And then the rest of the process is arranged by the research integrity officer at the institution. Initial inquiry, and then if there is enough to look deeper, it goes to investigation. And these are conducted by committee of scientists in that particular field. And the report is submitted to ORI for its oversight review. And we look at a lot more and review everything, evidence and scope and everything, and determine whether we want to make a separate public health service finding or not. If we decide to move forward with the finding, we reach out to the respondent for voluntary settlement. If the evidence is strong, we can convince the respondent that there is misconduct. Even when they see it, they sometimes challenge bringing up some procedural issues and things like that. Then it goes to a legal process. We work with OGC on a charge letter and administrative law judge proceeding. Sometimes it goes through full hearing, or it's with a summary judgment. And as you can see in this whole process, how many individuals are involved and waiting for the whole process, a lengthy process. As you see, inquiry based on our regulation is specified as 60 days and investigation 120 days, but it doesn't happen in that timeline. There's always a request for extension of that timeline. And when it comes to ORI, we also spend some more time. So this whole process is time consuming, and how we can reduce that lengthy waiting by new technology that can help actually to shorten this. So initial step, as we all know, we have to download or acquire the question data so we can save it and analyze later. But it has to be properly identified, because this is science in the context of, in a legal context, we have to have a complete identification of the data and everything else. So we download data from PDFs, Word files, and then from electronic devices. And some of the data, some types of data are deposited in data banks. An example here I am giving is protein data bank, where the crystallographic or other type of data is deposited. We have to download them and start analyzing this on the type of, and if it's digital evidence, we need to have specific information about the files, metadata, system data. So this is what I meant. We have to have downloading everything with proper identification. And the next step is looking through this data and what tools do we use? And we start with a human tool, and that's visual analysis. It helps a lot. It's a very important first step, and it has helped a lot in the past cases. And I'll show you several examples of visual analysis. But this is less efficient, time consuming, as we all know. Also, it needs confirmation to kind of convince the respondent and the judge. We also use some semi-automated tools that are inbuilt in some softwares, and I'm providing an example from Excel tools, and there are some PowerPoint tools and so on. We also use some forensic program like in-case analysis and features in other softwares. And I'm going to show you several examples. The initial slides will be visual analysis and how they helped. So examples, that's what rest of my slides are going to be, many of them. So here is, do you see my screen well? Yeah, I think so, yeah. So left is a figure from Nature Medicine, and the same author submitted an NIH grant application. I don't know if you can already see the similarities, but so, why is it not? We can see the same figure from Nature Medicine has been reused in grant application, but with slight manipulation of intensity and positioning of those cells. So this is kind of relatively okay, that if you stare at it long enough, you can probably see it. But I am saying there is more problem in the grant figure. I am saying these two are exactly the same cells reused. Not just that, these two are also the same cells that are reused. It's hard, right? It's hard to look and identify this. Can technology do this? I don't know. But with a little more work, we can actually convince the respondent, ALJ, and anybody the deeper hidden features that come up. So enhancing intensity and size adjusted, you see the similarity here. So another example where a small data set has been generated into a new image data. You can see the color-coded circles are exactly the same cells copied and pasted to generate this new image data. And these are probably possible to achieve without going through one by one visually. So I think that might be a simple thing for technologists to do. Light microscopic figure. Here is a figure 4 from a journal. Supposedly a pancreatic tumor injected with engineered bacterial spores to treat cancer. It's a big claim. This study went on and even submitted a FDA application for human trial, but this figure is falsified. You can see this part comes from a different image. And here, this is claimed to be hepatic tumor, presented as pancreatic tumor, a small piece of it formed the first control image, and then yet another one. So that one figure, again, was reused in many, many, many different grants and papers, and you see there's overlapping little portions were trimmed, manipulated, intensity manipulated, and flipped, and Even worse is like small portions in a different shapes put together for a new figure. I am just putting this out. This is how creative this happens. And the time I spent on this particular case, it's like days, I don't even remember. Then sometimes I had to print and flip and put it to see is it really, and that kind of thing. And yeah, I used quite traditional way of doing it, but I am sure you all can help us with this. This I am putting, this is another image that I had to use my visual analysis because this is a poor quality figures. All of them are like from poor PDF, and I don't know if automation can get here. This needed also a lot of days to look, and also the dates were very important in this case when the respondent said they lost their computers, everything during lab move, yet, but some 2003, 2006 figures appeared in grants submitted in 2011 and 10. So all that information helped get this case moved. Another type of falsification of Western blot is splicing in. Splicing in band pieces are not itself is a falsification, but when you have raw data and it's a beautification purpose that they are splicing, it's okay. But here, we had to look into it. So when you enlarge these Western blot images, you can see in these arrow marks, there is a splice mark. And when you, in the PowerPoint, when you get hold of each of these bands and pull them apart, you can separate them out. Not just separate them out, you can also see there is a band underneath. And this kind of indicates that this is not actual data that's put together, but someone is trying to fake data. On top of all these Western blot figures had also quantitative data in graphs. Bar graphs were provided, presumably collected from these Western blot band images. So there's additional falsification of numerical data. So when we went to look for raw data in the hard drive, there were files called libraries. There's no question about intentional falsification here. There's no raw data. Actually, this person, who knows where he put these libraries together from, and we could identify little bands that formed those blot figures. This is the extent that we have to go to convince. So not just microscopic images, not just Western blots, electrophysiological tracing. I'm talking about visual identification. Can you imagine looking through these? And in fact, this is supposedly generated from the equipment, electrophysiological equipment. However, as I marked here with the colored little demarcations, these little pieces were copied and put together to generate new tracings. And if you enlarge those portions, it shows the similarities. Can we really go through this one by one? We have, but I think with technology, we can shorten the time. So this particular kind of manipulation was in just single figure had 17 of these tracings out of 47 tracings in a single figure was generated by this, like this. And 4 such figures were included in 2 major journals, Science and JBC. It is a big problem. Another type of manipulation, it is painting in signals in figures. I am showing here an example of a microarray data figure, but this I see it in microscopic images like fluorescent, light microscopic, electron microscopic figures. People paint in signals or delete signals using Photoshop. Can we identify these? So I'm going to illustrate this. So this is a figure that appeared. This is the original data generated, but there is an insert to show that these metabolic genes are upregulated. You see the red, so I'll enlarge that to show. So here these two have signal. But then what we do is actually enlarge that portion that they are showing in the inset, and you can see those red signals are missing here. Sorry. So it is painted in, and it is common. I had another case where a fluorescent cell images had signals painted on them. So that's another type of manipulation for you all to think of. We also get graphs copied and reused. And here, this is a figure that contains 3 different graphs. But as you see, this first and third graph are similar. However, they are different too. So what did the respondent do? Added 2 new signals to fake it. These are 2 new signals. So it doesn't look similar, right? You want to disguise that similarity. But within our capability, when you enlarge these new and old signals, the new signals are a little smaller in size. This is our visual analysis. But you can go a step further by back calculating the data or reverse calculating. So it spits out numbers. Now you can't argue. The numbers are similar, exact numbers. So that's one way of doing it. And there are cases where we have to plot the notebook numbers to see if that's the published data. And that, I think that we can do. But this kind of manipulation, even an initial identification, needs a lot of time spent and visually identifying them. Okay, not just figures, images, but tables. When you don't have a software-generated software file like Excel file or so, when a table is published, there are other clues that this may be falsified. I'm not going into that detail, but here you see muscle weights from rats 1 to 6 in different columns. There's some abnormality in this data set. I want you to look at weight 3 and 6. They have more digits than other data columns. Not just that, they also end in 5s and 0s, indicating a calculation there. So here it turned out weight 3 is from average of weight 1 and 2, and the weight 6 is from average of 4 and 5. So here again, if you don't have original data, you do have a table that's questioned. This is a time-consuming way of doing it. Copied files. Can you identify copied files? The lengthy, tedious way is to open the file contents and looking at them. This is gene sequencing files. This is from a recent case of mine. So when the student was leaving the lab, the PI asked for raw data, and he said, yes, I will upload. And you can see different files, different file names. But if you open those files with the software, it shows exactly similar data inside. Metadata or generation date is in 2007. This is before the author even joined that lab. What he did, quickly copied somebody else's previous files in October 2010. So again, a tedious way of opening one by one to find copies, but I know I'll show you later in my slides how you can also do it with the software, but this is a tedious way, and this also needed further confirmation to demonstrate. This is about 149 files that were copied like this. Okay, so that's lengthy visual analysis combined with a little bit of technology. Here, I'm going to show you how we use forensic tools and software tools. This is again a spliced Western blot and image. This was in a grant and a paper, but institution also submitted PowerPoint files with exact figures. And many of you may already know that PowerPoint features, depending on how the respondent made the figure, you can actually use the PowerPoint features to show this. So I'm saying these 2 bands were derived from some unrelated experimental film, and this is from that. And so I'm going to show how I got there. So if you click on this band, can't see my, and go to format, and it's not coming. Oh, and then say crop. It brings that original film that this was cropped from. I can show that to each one of these other ones, and that's how we use PowerPoint features to identify. Sorry. Okay, so this is using PowerPoint features. We use Photoshop gradient map a lot, and I know some of you also use it. It's on our ORI website, thanks to John Creeder, who was a previous investigator in our group, and he has put these actions, Photoshop actions, online for everyone to use, and we do get a lot of analysis. People used it. So this is used when you cannot convince someone that bands are similar. This is a case where just individual bands were pasted on a panel of blood or image or some panel. And I say that these red ones are same, and the blue ones are same, and the green ones are same. But you will be surprised how many times respondents argue, no, it's different. Look at the cut up here and down here and things like that. So one way to bring out the similarities is to false colorizing these bands. It's better. Now you can see features of band structure, and this background is just from some blank something. But if there is artifacts, that also come up. Again, still they can argue they are not same, but you need to do some more work on it to exact similarity. And now it's difficult to argue, right? They are exactly the same bands that were pasted on. Okay, see the similarities in the band as well as the outside background. Flow cytometry data. These are graphs that are generated by the equipment. This is a very difficult data set to identify and to prove, because respondents bring a lot of technological explanations. However, through our own experience and working with flow cytometry experts, we found also came to know that overlap in area of sparse data is most significant indicator of those being copies of each other. As you see in green ovals, the dots are in the same places and they are similar. So in the same way, the bottom row shows in blue circles. And these are indicators of graphs being copied, but you cannot actually completely prove that. So using Photoshop overlay features, that's another feature that we use mostly for even Western blots and any other images. The overlay features, here I'm showing the graph 1 overlaid on itself to demonstrate the complete overlay will look red. When they are not completely overlaid, these dots will be black. So when overlaid on itself, it's red, and overlaid on this second graph, there's a lot of reds with some black. So adding additional data to disguise the similarities, same thing here. So this is a use of Photoshop overlay features. So how do we use inbuilt features in Excel? When we have the original Excel sheet, we can also do a lot with that software. So here, this data set is apparently hand entered, but when you put your cursor in that cell, I don't think this is hand entered. Okay, you all agree. But this is not the only issue with this figure. Experiment 1 probably is original data, but these were not. I can show you by going through this check formulas command. Formulas were used. without doing experiment and getting data. Formulas were used to derive those data. So that's one application through Excel sheets. I also use sorting data features in a recent case where you can see these are two data sets for a thesis, chapter 3 and chapter 5, claiming different experiments, different results, but this person copied data. So if you go and click on these columns and say sort data, Excel can sort data and you can see the similarities. The whole data set has been copied. Okay, so this is one way we use what is existing in the software. Okay, so if there's no software data, if there's no machine data, what do? Can you say that these numbers are fabricated? Again, one of our previous investigators in ORI wrote a statistical program called DigiProbe. The principle of this is that experimental noise is random, and it's hard to make up random numbers without a calculator by a human. And generally, human-generated number, they pay less attention, and the last digit, the rightmost digit, they tend to prefer certain digits than a randomly generated number. So the made-up numbers are not uniformly distributed. So this was from a case. The respondent provided a spreadsheet and didn't have printout from the machine. As you see, you can see, you notice here, the terminal digits prefer 0s, 2s, and 9s, right? So using this statistical DigiProbe program, it was the author tested the handmade no instrument tape numbers, suspected set of numbers, and numbers from actual machine data. So you can see the significant expression of high numbers of 0s, 2s, and 9s, highly significant at the last digit, whereas the machine data is all uniformly distributed. There is no preference. So it became useful for, in one of my cases, I suspected that respondents fabricated those numbers that she published. And you can see these files, the PMK, all these 6 data sets, I put through this DigiProbe software together with an exemplar data that I contacted the institution, got some machine numbers, and I put them through, and you can see it helped. So this person has actually fabricated numbers, and the machine numbers did not come significant. But with the statistical program, you cannot actually convince someone that, oh, these are in fact fabricated. But we had to do further analysis, and we did. NCase has saved cases for us. So here in this case, when asked, the same respondent from my previous slide. So the person said, I generated data, I collected data. This was about 293 data files we are talking about. I'm just showing a subset here. She said, I generated data days and days and days sitting through. And so she provided a disk with original data, but the disk was corrupted, couldn't open. We couldn't open, the software company couldn't open. So we don't know, we can't open and say that these are whatever. But looking at the file names, look at these file names. They are very different file names. Not just that, the generation dates are different. Dates, times, everything shows like she actually generated the data. So the committee at the institution did not make a finding because there is evidence that the data was generated actually. So when it came to ORI, we put it through NCase. NCase hash value came out. And I know you computer experts can tell what that means. And for us, I mean, so changing file names will not, or anything else outside the content of the file will not change the hash value, but If you change inside, it will change the hash value. For example, if you have this sentence, it's this hash value. If you put a dot inside the content, the hash value changes. So what does this say? These four are exact copies of each other because hash value is similar, and the same here. And it was true for many sets of duplicates or copied files. And this, then we had to go back to the institution, provide this evidence, strong evidence, and the committee agreed, and the respondent had no argument, and she was caught. So NCase saved this case for us, and there could be a lot more technological tools that we can use this way. So do we need technology? Yes, to improve efficiency and reliability. Efficiency, why? Because it reduces burden on the scientific community at large. Everybody, including complainants, respondents, reviewers, investigators. It protects the public. If we can do it faster, the fake data from the literature can be removed in a timely manner. It can protect public and prevent clinical studies taking off and killing people and things like that. And overall cost can be saved. So we need to work together, and that's why we are here. So that's about efficiency. Why reliability? I told you, this is science in legal context. We need to have this very strong, reliable way of doing things. So we need reviewable methods with description if possible, so everyone understands. Not sure if we can understand the whole computer codes. We have to have reproducible results to prove that this is in fact something, and possible to cover full scope without less false positive, which is, again, it's going to expand our time then. User-friendly if possible, and added capabilities on existing technical tools, and contemporary methods to keep pace with advancing technology, which we all know science is growing with in its own way with techniques used. What I've shown here is a very few that I handled, and there's more coming, and there's more that I have handled I couldn't show within this time. But we need to improve technology because falsification technologies are more, science technologies are more, and so on. So that's why we are here today, and this requires interaction and collaboration of professionals with different backgrounds and expertise. Scientists have to be involved, and computer personnel have to be involved, and that's why we are here. And thanks to Dr. Acuna for doing this, and we are excited. And I'll take questions if there's time. Thank you very much.